molecular methods that are often used in wine microbiology labs, and they include methods for DNA extraction, PCR, different kinds of PCR or polymerase chain reaction, and DNA sequencing, as well as proteomics. So one of the things that we would have done in the lab is a genomic DNA extraction from microbes in juice. And so we would have used a QIAGEN kit, which is a company that makes the kit. And there is a video that I posted in the lab resources on how to use the QIAGEN kit and what the workflow looks like. So at least you get to see it. But the basis of it is here. First of all, you would collect your sample. And again, usually it's juice, but it could be anything at all. It could be something more sophisticated like ground up leaves from grapevines or bark from vines or soil samples from the vineyard, you name it. But if it's anything complex like that, you would obviously have to separate the heavy stuff from the microbes eventually and then perform this lysis step. If it's just juice, we would put the juice in directly and lyse whatever microbes are in there. And so the lysis step really just involves treating the cells harshly with chemicals that break down peptidoglycan and proteins and cell membranes. So in this particular step, the net result is a solution that consists of a bunch of busted up bacteria and yeast cells, as well as DNA, RNA, protein, sugars, and lipids floating around in this matrix. The innovation here is that there's a column that you apply your sample to. Your lysis sample goes into this column. So the column looks like this up close. So your lysis sample goes into here. And basically the column consists of a matrix that will bind DNA, and everything else will move through into the waste here. The basis of the way the columns work is that they're typically hydrated silica. So you can see the silica atoms and then the hydration here. And then at some point in this binding process, you add a salt solution that replaces the water with positively charged sodium ions that can then bind to the negatively charged DNA. And so it captures that DNA. So as the DNA is being centrifuged through this tube here, the DNA will get stuck in the filter here. The rest of the material will pass right through. You can then wash the DNA. So again, the DNA would be stuck here, and you can put some wash solutions through that will just get rid of additional impurities and collect them here. And then lastly, you can take this filter basket, as we call it, put it into a clean tube, and then put some sterile water or something to then make the DNA dissociate and then pass into your tube. And so that's your clean, usable DNA. And so one of the things that we would have done with it in the lab is PCR. So this is a conventional PCR, or at least the outline for conventional PCR. And again, here's an example of a video on PCR that you can look at. And there's so again, lab resources on Polylearn that you can use to follow some of the links and study PCR. The basis of it is that you whip up your PCR reactions, and again, this video in particular tells you what goes into the PCR. It tells you what the thermal cycler, this machine here, does to the PCR reaction, and gives you a sense for how you amplify DNA using PCR. So you start with very few copies, and then PCR will allow you to create millions and millions of copies of those pieces of DNA. So the first thing that happens in this tube is there's a denaturation step where the thermal cycler here will raise the temperature of your PCR reaction, pictured here, and cause separation of the double-stranded DNA. The next step is annealing, in which you have specific primers that flank the gene of interest. And then the next step is a extension period in which the polymerase is allowed to add nucleotide bases and make copies of the DNA that's flanked by these primers. And then so this gives you a sense for the exponential amplification. Another version of PCR called quantitative PCR is pictured here. And so this is something that ETS Labs specializes in, and you can go to their webpage, which I've provided links for in the lab manual, they have this approach patented in which you can use these scorpion probes for quantification of spoilage microbes in a sample. And so the basis of it looks like this, where they have these specific scorpion probes that they've designed. And these probes can detect, they have different pools of probes that can detect Brettanomyces, for example, or lactobacillus or acetic acid bacteria or pediococcus. So they have different probes to detect different things. So if a winemaker is sensing some off flavor, they may report that to ETS labs, and then ETS labs can do specific scorpion probes to quantify and identify the potential spoilage microbe. ETS labs might also just do a full panel of these. It depends on what the winemaker is willing to pay for, because they're not super cheap. But anyway, so in this particular PCR, here's our scorpion probe, and here's our template. So again, imagine you've done your DNA extraction, as I showed you previously, from some juice or fermentation sample or whatever, and the first step would be the thermal cycler would heat denature the samples. And so again, you see the template denaturing, you see the scorpion probe denaturing as well. You can then cool and anneal primers. In this particular case, this region of the scorpion probe finds a target. And so again, if this was a Brettanomyces probe, then this region right here would be designed specifically to bind to Brettanomyces DNA. The next step is the extension phase. So the polymerase would come in and extend this sample, this DNA template, to make a copy of it. You would then heat, the thermal cycler would then heat denature, and you see the scorpion probe denature, and you see the other strand, the template strand, fall off. And then in this particular case, you cool it down again, and then the scorpion probe flips over and binds to a region of the newly amplified DNA. So this hunk of DNA right here in the scorpion probe has affinity for a region here that's been amplified. And so the cool thing about that is that, and that's why they call this scorpion. You can imagine this like a scorpion tail, how the probe flips over and kind of, that this would be the scorpion tail here. Anyway, so this region forms homology with the newly replicated strand here, and by keeping this quencher and this fluorescent dye separated, you get fluorescence emission. When the scorpion probe is like this, and the quencher and the dye are together like this, you don't get any fluorescence emission. So basically what the computer is looking for in the thermal cycler is looking for this event right here, the separation of the quencher and the fluorescent dye, because that will emit a ping of light that the computer will recognize. And so every time this happens, you get a ping of light. And so the computer will plot that as fluorescence versus number of cycles. So this is one cycle right here. And then the process would continue for 30 or 25 or 30 cycles. And so again, at the early stage, this is happening and fluorescence is being emitted, but it's not enough to rise above the background, and that's what this line is right here. This is just background. But eventually, enough of these reactions will happen in the sample that the buildup of fluorescence will start to become noticeable. And that's where you hit this CT value, this threshold, where the amount of fluorescence starts to rise above background. And then the computer will actually convert these fluorescence values into cells per milliliter values. Here's a no template control, so obviously the primers never find their mark because the mark doesn't exist in the sample, and therefore they never fluoresce above background. In so-called endpoint PCR, all you're doing is looking typically for presence or absence. And so here in lane 5 here, the DNA sample that you've put in here is not present the target is not present in this DNA sample, and so the primers never found anything, and so as a result, the PCR is unsuccessful. Well, it's unsuccessful because the template doesn't exist. In these samples, the target does exist, and as a result, you see a band. So again, the workflow: you purify DNA from the gel. You can then determine the DNA sequence, and then you can analyze the DNA sequence of this. So it's not just a presence or absence in endpoint PCR. It's also, what is this DNA and what's the sequence of it? So that can give us a lot of useful information about microbes in a sample and their identity. However, in this particular case, it's in quantitative PCR, it's identifying microbes, but it's also quantifying microbes. This approach does not quantify microbes. It just tells presence or absence, and then, like I said, you can analyze DNA sequences to figure out what they are. So this is showing you some of the factors that influence microbes in wine fermentation. And so they're broken into different categories. Microbes in the soil can influence wine fermentation, microbes on the vine, and the vintage and the climate of obviously the climate of the winery and so forth. There's a lot of anthropogenic factors that go into it as well. And so there's a lot of push these days to use genomic data in order to figure out what your typical microbial diversity is on the vines and in the soil and even around the vineyard. I'm sorry, around the winery itself. So this is just an example of that, but that's, so individuals have started using these big data set genomic analyses to figure out the types of microbes that are present on leaves and flowers and bark and in the soil to give themselves a better sense of what makes their wine unique. Is it something that's coming in from some of these other places, and therefore, if you know about it, maybe you can work it to your advantage. Or if there's something that's getting in periodically and causing spoilage, where is it coming from? And so if you have enough data about your surroundings using these genetic tools, you can actually figure out where the problem might be coming from. And so the workflow looks a little bit like this. I'm not going to give you a lot of detail, but I just want you to have the concept in your head, because there probably will come a time in the near future where this becomes more mainstream, and every winemaker is going to need to know about this. So it begins with, again, a juice sample, a soil sample, whatever, and you do a bulk DNA extraction from that. And you end up with a big ball of different kinds of yarn here. In each of these little strands, imagine being a complete genome from some organism in this mixture. Again, it's almost like throwing a bunch of clothes in a washing machine and then having to sort through it all. And obviously, you can do that if it's clothes in a washing machine, because you can pull everything out because you can see it. But here, it's a washing machine full of instead of clothes, DNA fragments that are not visible with the naked eye. So the way we do it then is a fragmentation initially, where we break the genome pieces into smaller pieces, and then perform the DNA sequencing. So you get sequences of all these tiny little fragments. This is sort of the black box part here, because sophisticated computer software then assembles these pieces and says, okay, here's a green piece, here's a green piece, here's a green piece, and then assembles those pieces into a long fragment. And so after doing all the gene finding and annotation and phylogenetic binning means you're just kind of dumping sequences that belong to the same organism into the same group, and then ultimately forming a genome out of it. So the genome that was in here in one piece gets broken up, sequenced, reassembled, and then basically compiled into one genome sequence. And so the cool thing about this then is you can not only identify the different kinds of microbes in a sample, but you can also go through the genome and get metabolic reconstruction. So I can tell, oh, this is a microbe that likes to produce whatever, diacetyl, and so, or this is a microbe that produces excessive alcohol because of such and such a defect. And so you can get a really cool metabolic reconstruction of the types of microbes that are coming in on the grapes or wherever. So here's another look at that. so here's our complex sample, and the the workflow can go in a few different directions. If you take this soil sample in this particular case, but again this could be a pile of grapes or who knows. One approach is to separate the cells from the matrix, and that includes just taking some of the soil or taking some crushed up grapes and plating them. Or it could mean just vortexing the heck out of it, putting the soil or the grapes into a sample, vortexing it like crazy, and then spinning down the heavy stuff, and then taking the supernatant. And then, so again, there's a lot of different ways to do that, but again, it's possible you could culture, it's possible you can physically separate soil and microbes, or at least most microbes from the soil, or whatever complex matrix. So in this particular case, now you have a mixture of microbes. You can do, if it's pure culture isolation, you can do single genome sequencing, for example, to figure out what individual microbes are. Or you can lyse all the microbes together and do this sort of high throughput sequencing that I showed you in the previous slide, which would then give you a fuller picture of all the types of microbes in there. Some individuals, and this is what I typically will do, is we'll take the complex sample and just directly lyse the microbes in there, and then and so again, that's just a sample prep issue. It's not. The end result is the same. You end up with a whole collection of the different types of microbes in a sample. Another thing that we can do, though, is RNA analysis, and this gives us an idea of the types of gene expression that's happening in a sample at a given time. And so imagine instead of a soil sample, this is juice from a fermentation from day 1 to day 12. And so each day you can pull out samples, do RNA extractions, and measure gene expression, changes in gene expression over that time frame, which would then give you a lot of information about the dynamics of the metabolism going on in your sample. So one of the things that we do for DNA sequence analysis is there's lots of online databases, and BLAST is probably the most well known. And so I have a workflow for this in your lab manual and some of the other materials. So I have a hypothetical DNA sequence as well, and not a hypothetical, an actual DNA sequence, and that you can use this workflow to identify it. So you would imagine that you PCR amplified that DNA sequence from some DNA that you purified from wine or from a pure culture, and then you subjected that PCR to that PCR reaction to DNA sequencing, and then you got a DNA sequence back, and then you use this workflow here, nucleotide blast. So I would click on nucleotide blast, which would then take me to a page like this. And then I can cut and paste any DNA sequence I have into this dialog box here. I can mess with my parameters if I'd like to and use different databases. I can tell the computer that my sequence isn't very good, so maybe I would do somewhat similar, or if I know my sequence is really high quality, I might pick this one. But either way, you can then click blast, which would then, after a period of time, bring you to this is just a cutoff. So, but anyway, follow the workflow in the lab manual and the workflow from the documents on Polylearn just to get a better sense of this. But they would bring you to an information page where you get a list of the top, a list of the top alignments, the microbes that most closely align to your DNA sequence, and then it'll give you very specific details about the actual alignment itself, such as the score, like how robust is this alignment. The expect value, which is what's the likelihood of your sequence being matched to this database sequence, is that it is random. And so in this particular case, the sequence that I uploaded in this case is 375 base pairs long, and so all 375 base pairs of my sequence matched the 375 base pairs of a sequence in the database. So that's 100% match, and that's why the expectation value is 0, because there's a 0% chance that that's random, that many bases had 100% match. And so you don't have to worry about these, but anyway, the point is that once you do this kind of thing, you can identify the DNA, and then in turn figure out what types of microbes are present, or or what type of metabolism they have. It depends on what gene you've amplified. Another thing I alluded to before was gene expression tools, and specifically proteomics. I talked about proteomics when I was referring to Brettanomyces and what changes, what physiological changes it makes when it goes into the viable but non-culturable state, or VBNC. And so I gave you some proteomics data. This is also a proteomics workflow. It's a little different than the one that I showed you previously, because the other one involved a special gel that you run the proteins on. In this particular case, called iTrack, the iTrack workflow is more advanced. And so in this particular case, you're using HPLC to separate the mixtures as opposed to a gel. But you're still using a mass spectrometer to identify the individual separated proteins. But the cool thing about this is that you can have different samples and extract proteins from those. So instead of DNA, now you're extracting total proteins. And then you can label those with a different dye. And so here's 4 different samples that I've extracted 4 different pools of proteins from, and then I've labeled them with 4 different dyes. And then I create my mixed sample that I run through HPLC and then the spectrometer. And the cool thing is that the software, once it's identified all the different proteins in each sample, the software will then tell you the different levels of protein in here compared to here, compared to here, and compared to here, which is great because then I can find out how much of a specific protein is present here compared to here, or here and here. So anyway, the point is that we can get a sense for the changes in protein profiles through the course of a fermentation, for example. And that would give us tremendous amounts of information about how metabolism changes over the course of a fermentation.